Hi, I'm Terrence Sejnowski, and I'm here in San Diego at the Salk Institute. I'm also on the faculty at UC San Diego across the street. And I'm going to be telling you today about a story to give us some insights into how the brain accomplishes working memory. Working memory has been studied over many decades. Recordings, for example, from both the prefrontal cortex and the parietal cortex by Pat Golden-Rakish and Richard Anderson have shown that there are neurons that are active during a delay period when a monkey has to remember a particular location or an object. And this delay period activity has been of intense interest to modelers like me to try to understand how the circuits in the cortex could be responsible for this elevated delay period activity. So the story I'm going to be telling you about today is only possible because of the fact that there have been major advances in machine learning over the last 10 years, which now have been used by researchers in not just my lab, but throughout the world to try to illuminate the complexity of some of the circuits that are found and some of the functions that are found in different parts of the cortex. So going first to some data. So this is a data repository that we took advantage of. That's the CRCNS NSF program, which recorded single neurons, spike trains, prefrontal cortex, dorsolateral prefrontal cortex of monkeys during a working memory task. So let's go through it. The monkey is first trained to fixate for 1 second, and then a cue comes up on either the left or the right side. And then after a delay period of a second and a half, another sample comes up that could be either on the same side or the other side. And if it's on the same side, the monkey is instructed to saccade up. And if it's not on the same side, then the monkey doesn't have to saccade. So this is a wonderful data set from hundreds of neurons recorded simultaneously during these different parts of the delayed matches sample task. And we're going to be analyzing these data along with data from our model to try to compare them. So we'll come back to this. If you go back 20 years, the dominant model for this delay period activity was called an Attractor network model. And this illustrated here, it was known that if you look at the connectivity pattern of the cortex, it's dominated by recurrent connectivity between the excitatory connectivity between the pyramidal cells located nearby. And that produces a lot of positive feedback. And then, of course, there's feedback inhibition, from the pyramidal cells, which helps maintain stability, because otherwise it would just blow up. And then you have inputs coming in that could activate a subset of the pyramidal cells. So this was this anatomy suggests that there might be a very simple explanation for the delay period activity, namely that you take advantage of the positive feedback to produce activity in a subset of the pyramidal cells. For example, if the inputs activate these yellow neurons, then they would be activated. So here's a pulse coming in, and with the right parameters, you can activate that subset. And that can be maintained in the absence of an input, but if you now give it a negative pulse, that can turn it off. So that will maintain activity in this upstate for the subset that have been activated. Now, there's a couple things that were a little bit concerning about this class of models, the Attractor models. First of all, there are dozens and hundreds of parameters here that you have to determine, depending on the detailed model. For example, if you have Hodgkin-Huxley model neurons, then there's hundreds of ion currents and time constants that have to be specified. And so you're searching through this very high dimensional parameter space. And of course, you know, if you search long enough, you find a set of parameters that actually gives you this performance. And there's something unsatisfying about that, because of course you haven't really searched it completely. Maybe there's another model that uses the same components and maybe even the same connectivity. And how are you going to find it? So that's what I'm going to be focusing on today, taking advantage of some of the modern tools and techniques from machine learning. So here's a model that was developed by Robert Kim, a graduate student in my lab. And it starts with a recurrent neural network of a few hundred units that are connected up initially randomly. 80% are excitatory and 20% inhibitory, which is the proportions found in cerebral cortex. And they were given inputs, and then there was one output. So, this, in fact, this one, if there's one input that is being fed into all of the neurons and through weights that are going to be adjustable by a learning algorithm, and then also we can see that the output is going to be driven, and that output is going to be compared to the correct output, and then on the basis of error, the weights in the recurrent network are going to be updated, and that's going to continue until it converges. So that's a typical machine learning task, but the difference here is that we're going to be doing it with spiking neurons, because traditionally these reservoir networks are use continuous valued units, a rate code. But real neurons, of course, spike at irregular times. And you saw in the previous slide recordings from the prefrontal cortex how irregular that they were. So here is now on the bottom an example of a trained network that is responding to the 4 different conditions. So remember that if the Q and the sample were the same, then it should go up, and so that's shown here. But if they were down, which means that they were on the other side, then it should also go up. And so these are the correct trials, and here are the incorrect trials. That's to say, the ones where you want a down response, and it's when they're opposite. The Q does not match the sample, and what you can see here is that there's a lot of variability occurring in the individual neurons, both excitatory and inhibitory, and especially elevated activity during the delay period, which is in between the 2 stimuli, and so now we've created a network. We don't know how it works, but it's somehow able to reproduce both the input-output behavior of the monkey, but also we have access to the network. So we can go in and examine it with much greater detail than we can go into the brain. So in the brain, we can record from a few neurons, and you can you can distinguish between the excitatory and inhibitory neurons, maybe what layer they're in, but you really can't access the connectivity, the specific connectivity of those neurons, which is what we could do here. We could do that for every single input, for every trial, and that's what we're going to do now, is dive in and see if we can figure out the algorithm that this network has discovered. So we need some metrics, and so here's one of the important ones that we're going to use to both analyze the recurrent network model, but also compare it to the recordings. And this has to do with autocorrelation. So the first step is to bin in 50 millisecond intervals the spikes, the number of spikes for each trial. So here's 50 trials, and each dot is a spike. And you can see that, and sometimes there's no spike, sometimes there's 1 spike, sometimes there's 2, and that is illustrated below with a gray scale. So we can see there's a lot of variability, and that's what you see in recordings. And now what we're going to do is to correlate 2 time points, like for example, the first and the second, separated by 50 milliseconds, and we're going to average that over the entire 1 second time course. And that's going to give us an autocorrelation value that you can see up here, a little bit of 0.2. And then now we do that with 100 millisecond delay and then 150 millisecond delay and so forth. And we get a curve in which the autocorrelation is decreasing. We can fit that to an exponential with a time constant of 177 milliseconds. So that is a way to understand something about the persistence of activity within the recurrent network. Because of the fact that they're talking to each other, they will maintain a memory of something that came in 177 milliseconds earlier. Okay, so that's now something we want to apply to both the experimental data and to the model to see whether there's a similarity. So first of all, upper left corner, let's look at the baseline firing rate, and we can see that both the in the monkey cortex and in our model, trained model, the average firing rate is about 5 hertz, which is varies obviously up and down during the task, but it's in the right ballpark, similar variance. And now let's look at the autocorrelation of time constants with the time scale. And first of all, the untrained network down here, as you can see, has a very short memory, 7 milliseconds. And that's because the connections are random, and there's no reason why it should have recirculated spikes. If there's random connectivity, especially with inhibitory neurons that are going to be activating to reduce the activity. So, but if you compare that to the trained network, you can see that there's a much bigger spread of time constants, and in particular, the median is about 122 milliseconds, and that's comparable to what you see in the data from the dorsolateral prefrontal cortex. And if we split this into two, the shorter on the left and the longer on the right, and then just plot those the way we did in the last slide, we can see that the short time constants average around 68, 69 in both the experimental and the model, and similarly for the long time constants, 133. So this is actually a very good quantitative comparison. So it means that there's something that may, it could have some similarity, but we're going to need much more evidence in order to be able to conclude that. But at least I think we have some reason to suspect that the network has learned something about persistence. Now, the penny dropped when we started looking at the connections between the input and the inhibitory neurons, which is not expected. We expected that they would be connected to the excitatory neurons, because that's what our models, the Attractor models, told us, that the inputs were to the excitatory neurons that were going to go into the upstate. But no, it turns out here that the strong connections are to the inhibitory neurons. Okay, that's interesting. But even more interesting is the fact that there are two populations of inhibitory neurons. There's one subpopulation which received a strong positive input. They were selectively activated by the input, and then there's another set of inhibitory neurons that were not, and you can see that it was reflected in the weights. The weights were positive to the first subpopulation, or negative to the second subpopulation. So this is telling us that there's two subpopulations that have mutual inhibition between them, and this is something that we confirm by looking at the detailed connectivity. So that's a clue, and then we started manipulating the network. And by the way, this is incredibly important. It's something that we can begin to do actually in experiments. We can activate neurons with channel rhodopsin, or we could reduce activity with these optical techniques for manipulating the activity. And so we did that. We could either increase, in this case, through increasing the strengths of the connectivity between the two subpopulations, or decreasing them. And what you can see here is that if you increase the strengths, both the time scale and the performance actually increase a little bit, but if you decrease the strength, the time scale and the performance drastically fall, suggesting that these mutual reciprocal inhibition between the two subpopulations may be critically important for the delay period behavior, the performance, being able to maintain activity. We were able to explore both the connections between the subpopulations and also the connections within the subpopulations. And it turns out that the connections between the subpopulations were significantly stronger, and the numbers were significantly greater. And if you look at the task accuracy depended more on the reciprocal connections than it did by changing the strengths of the connections within the inhibitory subpopulation. So this is now the smoking gun, and it gives rise to a cartoon shown here about what might be going on in this network and might be going on in the cortex. So here is the model. Now shown here on the left is the input, and as I was telling you, if there's an input that is most strongly connected to one subpopulation, it will turn that on, and this subpopulation will turn off the other. So it's a flip-flop. Either this subpopulation is on or this one. And because of the fact that there's inhibition between the inhibitory and the excitatory neurons, the one subpopulation that is activated will turn off the excitatory population that it's paired with, and the disinhibited excitatory neurons you see on the bottom here are activated. So we have, during the input, we have a subpopulation of inhibitory neurons that are activated and another subpopulation of excitatory neurons that are activated, but there's no, as you can see here, there's no direct connection between these two, but it's always through the inhibitory, reciprocal inhibitory subpopulations. And what happens now is, because of the fact that that this is stable, you can turn off the input and in fact achieve an even greater firing rate, which will be maintained during the delay period. So this is the motif now that we think is responsible. And interestingly, other groups that have studied a completely different set of problems having to do with decision making, how do you decide to do something, if you have choices, have come up with a very similar kind of a reciprocal inhibitory motif. And so what that raises the possibility is that the very same motif, depending on how it's connected to the output and the excitatory subpopulation, that the very same motif could be used for both, which would be very economical, and also tell you that that there's not separate modules for decision making and for maintaining activity, that they're one and the same. Okay, now we'd like to have some more evidence, right, that this is actually going on, and so we found it in trial-by-trial variability. So if you record from a single neuron, say, during multiple trials, here's 5 trials, and you just look at the pattern of spikes across trials, there's 2 possibilities. One is that, we know that the interval, that the firing times vary from trial to trial, in this case, early and late on trials 1 and 2, but there's only 2 spikes. And sometimes it's 3 and sometimes it's 1, but there's very low variability in the total number of spikes. The variance is 0.5. And this could be quantified with something called the Fano factor, the variability in the number of spikes. Now, high variability would occur if, for example, that same neuron was highly active on one trial and maybe very little activated with, say, 1 spike or none on other trials. And so the question is, what's the Fano factor then? First of all, in the dorsal lateral prefrontal cortex. And here's the short and the long time scales that we discussed earlier. And what you can see is that the long time scales here tend to have much higher Fano factors. Up here in this layer 6 is very high. So this, by the way, this particular diagram had a Fano factor of 5.5. So we're dealing with this green condition here in the dorsolateral prefrontal cortex, highly significant between these two groups, the short ones and the long ones, suggesting it was the long ones that are really responsible for the behavior. And here now are the analysis for the recurrent neural network, and you can see that there's a similar excess of long, high Fano factors here in the long timescale group. And it's also reflected in the autocorrelations as well, and we tested both. And it's clear now that there's a very nice congruence here between real neurons and the variability in those neurons within the by the way, I should have said this is within the subpopulation of inhibitory cells recorded from the dorsolateral prefrontal cortex. And this recurrent network here as well. So this is now, again, this is, I think, pointing us in a direction which is revealing something that I think may turn out to be a real insight. And I have to say, it's not my insight. This insight actually came from the machine learning algorithm, right? The machine learning algorithm hit upon this particular network connectivity. And by the way, we ran the algorithm many times on many networks, and they always came up with the same solution. So it suggests that in some ways, this may be the most direct or simplest way that you can implement the delay period activity that has to be maintained, some pattern of activity maintained across time to be able to solve the task. And it's, I think, particularly interesting that the inhibitory neurons are involved here because they have been neglected. Everybody knows that they're there to damp activity, but now we know they have many other. There are many types, and they have many different functions, and so this may be one of them. So we wanted to play around with this in the model. It's one of the things you do with the model, by the way. It's very easy to change the strengths and cut connections and so forth, and very difficult to do that in vivo, of course. But this at least gives us more insights. So here's what we did. We trained up a network. We looked at trials, and I've already showed you here the high variability in terms of the number of spikes across trials. And now what we could do is strengthen or weaken the connections within the subpopulation. And what you can see here is that as you strengthen those reciprocal inhibitory connections between in the subpopulation, you can see that, between the two subpopulations, right? There's a reciprocal connectivity between the subpopulations. You can see that there's a lot more failures here, which means that the Fano factor has gone up, and that's shown here on the bottom, times that both the Fano factor goes up with strength, it goes down with reducing the reciprocal inhibition between the two subpopulations, and the time scale also plummets. It goes up and down by a factor of 100 milliseconds here. So this is, again, something that we are able now, because if you just look at the connections and so forth, it's hard to really understand just from the connectivity diagram what's really going on. But now we were able to zero in on a particular submotif that is responsible. Now, just to show you, that this is we need controls. I mean, the question is, what if you manipulate the strengths of the other connections in the network, right? And so here we have connections between the inhibitory and excitatory connections, the excitatory going to inhibitory and excitatory going to inhibitory. You can vary those up and down by 30%, and the Fano factor does not change. It really is strongly affected by the inhibitory connections between two subpopulations. And these two subpopulations are really the ones that are controlling the excitatory cells that are producing the output, right? So, and by the way, this is it's a little bit, maybe even a lot of a surprise, because everybody thinks that 80%, so these excitatory neurons are really doing the hard work, the heavy lifting. They're the ones that are communicating long distance, and they're the ones that are making decisions. No, in this network, it's the relatively smaller subpopulation of inhibitory neurons that are making the decision. And they're sending the troops off to battle, right? The excitatory neurons are getting their they're orders from the inhibitory neurons. This tells us that these inhibitory neurons might be actually even more important than we imagined. Now, there are many types of inhibitory neurons, and we focused in on two of them. And here's the premise. The premise is that we know that these delay period activity neurons are found high up in the hierarchy, parietal cortex and prefrontal cortex, and are not found in early stages of sensory processing at the bottom of the hierarchy. And so the question is, if we look at the subpopulations of inhibitory neurons, and the two we focused on here were the parvalbumin positive basket cells providing the feedback connections, strong inhibitory feedback to the pyramidal cells, and the somatostatin inhibitory neurons that are primarily activating other parts of the circuit, and also have strong inhibition onto the parvalbumin neurons. So there's additional complexity here, but those populations have been measured in a wide range of cortical areas, and they've been put into a hierarchy here with the early sensory areas and the somatosensory, the auditory and the visual system on the right, and then high up in the prefrontal cortex and the infratemporal cortex here on the left, orbital frontal and infratemporal and insula. And so if you look now at the parvalbumin density of inhibitory neurons, which you can see here that it's very high in the sensory areas, the early sensory processing areas. But if you go up to the prefrontal cortex, it drastically falls by a factor of 5. And interestingly, if you now look at the plot of the somatostatin positive inhibitory interneurons, they are very low in the early sensory processing areas, and they are very high. They reciprocally going up as the parvalbumin is going down. And so this really implicates these neurons. These could be the subpopulations of the somatostatin neurons. Would be a good place to look if you wanted to see which subpopulation was responsible for making these decisions and maintaining delay period activity. Okay, well, where does this take us? So I've shown you one of many examples now in my lab and elsewhere of many groups that have now applied these machine learning techniques to create networks and then to compare them directly and quantitatively with neural circuits and also with the firing patterns. And most notably in the visual system, there's a hierarchy from the primary visual cortex at the bottom up to infratemporal cortex through there's several dozen cortical areas that are responsive to different types of visual inputs in this hierarchy. And Jim DiCarlo, amongst others, has shown that if you train up a deep learning network on object recognition, that the patterns of firing of the neurons in different levels of the hierarchy are very similar to those that he records from monkey visual cortex in these different levels. And most recently, he's shown that if you add recurrent connections, feedback connections from a higher level to a lower level or within layers, which of course we know is in the cortex, you can get even better comparisons. And so this is great because it's before we kind of suspected that the hierarchy had something to do with perception of objects, but now I think we have much more confidence because we can create a network that actually has performance that is comparable to that of humans in terms of recognizing, for example, 10,000 different categories of objects, right, from millions of examples. And so now we can confirm that those properties that we see in these different cortical areas actually have the properties that you need in order to be able to do object discrimination. And if you're interested, I told the story in a book I published, The Deep Learning Revolution. The story here really goes back to Hubel and Wiesel. The network I just showed you, the convolutional neural network, was inspired by their recordings from visual cortex and then by Van Essen and others who recorded in the higher visual areas, and then Charlie Gross, who recorded up in infratemporal cortex. And so all of that went into developing this new class of networks called deep learning networks, which have had enormous success in artificial intelligence. And now, as those networks have been refined and expanded, the latest versions now that have been trained have a billion connections, a billion weights in them, which is, they're becoming not quite up to the level of the cortex. There's about a billion synapses in a cubic millimeter of cortex, so they're pretty far away from building a real brain. But now that they've been applied to hundreds, thousands of applications and have had great success, especially in natural language processing, language translation. And so now, as we understand more and more about these networks, as they become more sophisticated, for example, the latest generations have included attention, which has had a huge impact in performance. And as we add more bio-inspired mechanisms, we see that the improvement performance can now be taken back and give us insight into biological mechanisms. And this reciprocal going back and forth wasn't possible before in traditional AI, which was based on logic and rules. But in my book, you can see the tagline, Artificial Intelligence Meets Human Intelligence. And if you don't have the time to read the book, there's a shorter highlights in a paper I published last year in the preceding National Academy of Sciences on the unreasonable effectiveness of deep learning and artificial intelligence, which sort of gives us a guideline into what to expect, both in terms of the past and how we got here, and also some really exciting new mathematical advances that have occurred by mathematicians analyzing these networks, telling us, mathematically why they behave, because otherwise it would be a paradox. Okay, so that's the end of my talk, and I want to give credit to Robert Kim, who did this one of many projects in my lab that was really path-breaking. And thank you very much. As the sun sets over the Pacific, I say adieu.